Assalamualaikum warahmatullahi wabarakatuh. Good afternoon. Today I will deliver about the vitamin C metabolism, and it's our agenda for today's learning. Okay, vitamin C has the other name ascorbic acid and belongs to the water-soluble vitamin group, and the vitamin C is the strong reducing agent. Humans, primata are unable to synthesize vitamin C endogenously, so we can say the vitamin C is one of the essential dietary component like the vitamin E, vitamin K, vitamin E, but not for vitamin D because our body, especially our skin, can synthesize vitamin D cholecalciferol because in our skin has 7-dehydrocholesterol, and when the 7-dehydrocholesterol get the exposure UVB, so it convert to secosterol, and when the secosterol get the thermal process, the secosterol converts to the cholecalciferol, so we can synthesize vitamin D endogenously. Vitamin C is required for the biosynthesis of collagen as reducing agent, L-carnitine as coenzyme, catecholamine as coenzyme, and certain neurotransmitter. Coenzyme, co means assist to help the enzyme working. Vitamin C is also involved in protein metabolism, an important physiological antioxidant, and has been shown to regenerate other antioxidants within the body, including vitamin E or alpha tocopherol. And the sources of the vitamin C are the citrus group, like the orange, lemon, uh, and then the tomato, broccoli, eggplants, cucumber, chili, blackcurrant, strawberry, etc. The role of ascorbic acid in cellular metabolism can be accounted for by its reducing properties to protect cellular components from oxidative damage. It acts as a scavenger for oxidizing free radicals and harmful oxygen-derived species like the hydroxyl radical, the hydrogen peroxide, and reactive oxygen singlet, and and the free radical can damage our cells, our tissue, and our DNA. The total body content of vitamin C range from 300 milligrams, and its number uh, is at near scurvy until to about 2 grams. High levels of vitamin C are maintained in cells and tissues and are highest in the leukocyte white blood cell. And the leukocyte is one of the immune function. Why the highest in the leukocyte? Because the leukocyte will attack the bacteria or virus with the burst oxidative, and when the leukocyte attack a bacteria, the leukocyte will throw the free radical to bacteria or virus. So the reactive oxygen singlet and free radical will be formed through this process. So the vitamin C must donate the many more the electron to convert the free radicals resulted to the inactive species. So vitamin C can protect our body, our cell from the oxidative damage. A is adrenal glands, pituitary gland, and brain. Vitamin C can protect the brain because of the neurons in the brain have higher rates of oxidative metabolism and contain some of the highest concentration of vitamin C. Intracerebral vitamin C provides protection against oxidative stress and glutamate toxicity and supports peptide amidation, myelin formation, synaptic potential, and catecholamine synthesis. The intestinal absorption of vitamin C is regulated by at least one specific active transporter, and cells accumulate vitamin C through a second specific transport protein. Based on the in vitro studies have found that oxidized vitamin C or the hydroascorbic acids enter cells uh, through some facilitated glucose transporter and is then reduced internally to ascorbic acid. Okay, so the hydroascorbic acid is the form of vitamin C, oxidized vitamin C. Okay, you have to memorize it. Ascorbic acid is the form of vitamin C in the reduced vitamin C. Meanwhile, the hydroascorbic acid is the oxidized vitamin C. Okay, this is diagram for the cycle of the ascorbic acid and the hydroascorbic acid. Ascorbic acid is the reduced form of vitamin C, and the oxidized form is the hydroascorbic acid. Can be reduced back to ascorbic acid by a glutathione enzyme because it's thiol group, and the ascorbat is also involved as reductant to reduce back tocopheroxyl radical to tocopherol. This vitamin C content of food might be reduced by prolonged storage and by cooking because ascorbic acid is water soluble and is destroyed by heat, steaming or microwaving myelination, cooking loses. Fortunately, many of the best food sources of vitamin C, such as fruits and vegetables, are usually consumed raw. The vitamin C content of food is too strongly influenced by season, transport to market, length of the time on the shelf and in storage, cooking practices, and the coordination of the water used in cooking. Cutting or bruising of produce releases ascorbate oxidized, so balancing techniques in activate the oxidized enzyme and help to preserve ascorbate. Lowering the pH of a food will also achieve this, such as in the preparation of sauerkraut, and in contrast, heating and exposure to copper or iron to mildly alkaline condition destroys the vitamin, and too much water can leach it from the tissue during cooking. Okay, these are the source of the vitamin C in the selected food and the plant sources. Second one, the dietary supplements. The supplements typically contain vitamin C in the form of ascorbic acid, which has equivalent bioavailability to that naturally occurring ascorbic acids in foods such as orange juice and broccoli. Other forms of vitamin C supplement include sodium ascorbate, calcium ascorbate, other mineral ascorbates, ascorbic acid with bioflavonoid, and combination products such as ester C, which contains calcium ascorbate, dihydroascorbate, calcium threonate, sironate, and disonate. In one of the reasons, ester C and ascorbic acid produce the same vitamin C plasma concentration, but ester C produces significantly higher vitamin C concentration in leukocytes 24 hours after ingestion. Vitamin C and metabolites like the one supplement ester C contains mainly calcium ascorbate and includes small amounts of the vitamin C metabolites, the hydroascorbic acid, calcium threonate, and trace amounts of cyanate and lisonate. Although these metabolites are purported to increase the bioavailability of vitamin C, the only published study in humans addressing this issue found no difference between ester C and commercially available vitamin C tablets with respect to the absorption and urinary excretion of vitamin C. Ester C shouldn't confuse with ascorbyl palmitate, which is also marketed as vitamin C ester. Ascorbyl palmitate is a vitamin C ester. In this case, vitamin C is esterified to the saturated fatty acid, like the palmitate acid, resulting in a fat-soluble form of vitamin C. Ascorbyl palmitate has been added to a number of skin creams due to interest in its antioxidant properties as well as its importance in collagen synthesis. Although ascorbyl palmitate is also available as an oral supplement, most of it is likely hydrolyzed to ascorbic acid and palmitic acid in the digestive tract before it is absorbed. Ascorbic palmitate is marketed as the vitamin C ester, so we cannot say it ester C.